Thank you, Dror, for the invitation. So now for something completely different. So I appreciate having the chance to speak in the Chabner colloquium. It's always an honor to be able to have a chance to speak here. And what I'd like to do, really for I guess this is the last talk of the afternoon, is to give you a flavor about what genome scale studies of prostate cancer have been teaching us, not just about the disease biology, but potentially new ways of thinking about approaches to chemoprevention and eventually precision medicine. But I will warn you that unlike what Matt just told you and Mary Ellen and Rick, where the advances that we're heading towards better and better treatment, I don't have a new drug and a Kaplan-Meier curve to show you, so maybe I need to eventually shift gears and do more of that so I can feel like I'm helping patients more. But I think there's some very interesting observations that have come out of genome genetic studies of prostate cancer. So you can, this is one of these things where you can put your pencils down. This won't be on the test, but hopefully it'll be interesting. Okay, so in the era of oncology that we're now living in, we increasingly are realizing that the path to more effective therapeutics can be schematized, as shown on this slide, where rather than purely thinking about cancer based on the organ in the body in which it arose, we start to think about it from a genetic or molecular standpoint, and we want to identify dependencies that are operant that might lend themselves to a vulnerability using a targeted therapeutic. And if we get this right, we can achieve sometimes very impressive tumor responses. But unfortunately, particularly if these are single agents, the tumor responses are invariably transient. So one of the lessons that's been taught is that we need to achieve higher order combinations, and the principle here is that these combinations should ideally be derived both from larger scale knowledge of tumor dependencies, for example, enacted by recurrent genetic alterations that are drivers in particular cancer types, or knowledge of therapeutic resistance. And so the thinking is that if we can come up with principled therapeutic combinations, we will be more likely to move towards durable control of many types of cancer. And in prostate cancer, as you've been hearing, one can actually look at this tumor as though the glass is half full. Because if we think about knowledge of tumor dependencies, we've of course known for a long time that the androgen signaling axis is a critical dependency. As Matt just showed, the development of several rational therapeutics that target this blockade has emerged, and now we're starting to see even additional examples with XL184, et cetera, targeting larger sets of targets around the biology of bone metastases, et cetera. And we can achieve quite impressive responses with some of these agents. And so now the question is, can we move to higher order therapeutic combinations? And particularly, can we learn about additional dependencies, for example, by characterizing the genetics of prostate cancer so we can identify new biological processes that perhaps we didn't know about before, but might offer up novel therapeutic avenues. So our group has been working for several years now on characterizing the genomes of prostate cancer, and there have in fact been some very interesting observations that I think have lent themselves to further study. So I'm going to assume that this audience knows that cancer can be thought of as a disease of the genome. I'm not going to go through the various categories of genetic alterations or the technologies to discover them. I'm going to assume that people know that that's happening and it's powerful and it's exciting. I'm going to cut right to the chase of what we've been learning and go to this graph here, which describes essentially the mutational landscape of at least aggressive primary prostate cancer. This was a study of approximately 116 or so primary prostate tumors. The Gleason grade was usually 7, 7, and 8. There were maybe a small number of 6s, but these tended to be larger tumors, so that they came from prostatectomy, radical prostatectomy specimens. And what we're looking at is the distribution of several genes, mutated genes that were deemed significantly mutated in this particular cohort. And we're looking at the frequencies on one hand and the sort of a significant score on the other hand. And these are the overall background mutation rates for the tumors, which I'm not going to get into now. But the point of this slide is to show a couple of things. One is that when you look at the mutational landscape of prostate cancer, you realize that actually even in this initial view, you can see that it follows what we call a long tail distribution. So there are a smallish number of genes that are highly recurrently mutated, meaning greater than perhaps 10 or so % frequency, but most of them are mutated at much lower frequencies. And this, more or less, this long tail distribution is the rule for mutated cancer genes, driver genes in cancer. But what was interesting is, although we did see some genes that we recognize from tumor biology, for example, p53 being reasonably significantly mutated, p10, et cetera, these are well-known tumor suppressor genes, there were some surprises. So, for example, the gene that went right to the top of this list is a gene called SPOP, which is a gene that encodes the substrate recognition motif of a ubiquitin ligase. So the ubiquitin pathway in cells is sort of the garbage dump. If you add ubiquitin groups, polyubiquitin groups to proteins, it sends them to the garbage can. It tells the cell they don't need it anymore. In fact, it goes to the proteasome. So SPOP, it recognizes substrates and then essentially directs the ubiquitin ligase activity to add ubiquitin groups to these proteins. And what was interesting was that the mutations that we saw in SPOP, they exquisitely decorated the substrate binding pocket of the so-called math domain of SPOP, which is responsible for binding various types of substrates. And the elevated mutation rate was validated in several independent cohorts. So this was a very interesting set of observations because they clearly seem to be predicted to disrupt substrate recognition in some way. But on the other hand, this was really not something that was known to prostate cancer biology before. So as you've heard, a lot of the therapeutics have been directed at the androgen signaling axis for very good reason, but here now was potentially an entirely different cellular machinery that we could think about targeting for the first time. Recently, just briefly, when one compares the frequency of SPOP mutations with other genetic alterations that are highly recurrent and of interest, one sees that SPOP mutations, there's two properties that are of interest. First of all, they never, at least in our hands, co-occur with ETS factor rearrangements. So these are prostate tumors that have the TMPRSS-ERG fusion. About 40% or so of these had that, as expected. But the SPOP mutations were always separate from those, and we've seen that in every cohort that has been looked at. But they do co-occur with some other genetic alterations, in particular deletions, focal deletions at 5q21, which happens to include a gene called CHD1. CHD1 is a gene that regulates compaction of chromatin, so it seems to be involved in chromatin biology, which of course many of you will know that mutations in various chromatin regulatory factors have been one of the most striking discoveries of cancer genome characterization over the past several years. So we have evidence of that biology happening, but in particular in this subset of prostate cancer. So essentially, SPOP mutations seem to be genetically, they were defining for us a genetically distinct subtype of prostate cancer that is completely different, at least at a genetic level, from those characterized by ETS factor rearrangement. So the other interesting aspect related to this subset of prostate cancer is that it does seem that these mutations are driving tumorigenesis. So this is actually very recent data that done by a postdoc in the lab, which is essentially modeling proliferation of prostate epithelial cells that contain either wild-type SPOP or various prostate cancer associated SPOP mutations. Incidentally, this particular methodology uses prostate epithelial cells that were cultured using a new way of culturing cell lines using a fibroblast feeder layer treated with a Rho kinase inhibitor. This is actually something that has become of great interest because it seems like this is a way to grow cells indefinitely in culture. So there's a lot of interest, as you might imagine, to use this to grow out new cancer models. In this particular case, we grew out normal prostate epithelial cells, but it turned out to be a very robust model to assess the phenotypic effects of SPOP mutations. And you can see that the boost in cell growth for all of the prostate-associated SPOP mutations, but interestingly, mutations in SPOP that occur in endometrial cancer. So it turns out that endometrial cancers also have SPOP mutations at about 8 to 10% frequency, but the location of those mutations is completely different. They almost don't overlap at all with the prostate cancer mutations, and indeed, the two that we tested really don't have a substantial growth phenotype when you compare it to the SPOP mutations in a prostate epithelial cell proliferation assay. So it really does look like, number one, in prostate cancer, these mutations are driving the proliferation of prostate epithelial cells, and number two, this is a potentially new category of cellular process that we can start to mine for targeted therapeutic avenues. Very briefly, if we compare, so there have been a variety of studies now done on the genetics of prostate cancer using massively parallel sequencing technology. This was a review by Haley Hieronymus and Charles Sawyers that compared our study, which was done in primary tumors, to that of a group from Arul Chanayan's lab that looked at metastatic tumors. These are actually mostly soft tissue mets. These really weren't bone mets, but what you can see grossly, this is chromosomal copy number data. The primary tumors in general are much quieter from a genome derangement standpoint, at least as measured by chromosomal copy number data, than the advanced metastatic disease is. But when you look at the genes that are significantly mutated, there's actually quite a bit of overlap. There are some that are distinct in the metastases, but there's quite a bit of overlap. So there's a growing knowledge of the landscape of genetics that will hopefully give us many new targets to mine. So what I've told you thus far is when you go back to this schema, one of the new areas of knowledge that's come out of cancer genome sequencing is the idea not only about SPOP and ubiquitination, but in general, the idea that protein homeostasis could be a therapeutic avenue in prostate cancer in a way that we haven't thought of before. Now, protein homeostasis per se is not new to cancer. Many of you will know that bortezomib in multiple myeloma is a proteasome inhibitor, but bortezomib has never had any obvious efficacy in solid tumors. However, there are other protein homeostasis drug targets and therapeutics that are in early clinical and preclinical development, so certainly this will be something to consider in SPOP mutant prostate cancer going forward. Now, for the rest of my talk, I'd like to turn the page a little bit and sort of step back and point out that when you sequence cancer genomes and cancer exomes, you have, of course, the proximal opportunity to discover recurrently mutated genes, which is, of course, really why we want to do it. But you also have the opportunity to understand the evolutionary history of that cancer. And, you know, I've often thought when I open up a journal, Science or Cell or what have you, and I read a cancer genome sequencing paper and they're talking about cancer evolution, I think to myself, well, they didn't find anything interesting, there's no new interesting genes, so they had to talk about tumor evolution. But it happens in prostate cancer that there is quite a striking architecture in the genome that really we couldn't ignore, and it really has taught us something about evolution. But I think at the end, there are some very interesting sort of provocative questions about what this could mean for prevention and treatment for prostate cancer. So I'd like to give you a bit of a glimpse into that story. So it does happen because we're sequencing the entirety of an exome of a genome that you can infer quite a bit about the mutational history of that particular cancer, the clonal history, you know, is there heterogeneity, are there major subclones, minor subclones, the diversity of mutations, and whether or not there were bottlenecks that were induced by treatment. So these are all areas where one can potentially learn. And so the questions that arise from those types of studies are, number one, are there specific evolutionary processes that gave rise to individual cancers, and what are the implications for tumor biology and treatment? And as I mentioned, there were very interesting insights that came out of analyses of prostate cancer genomes. Now, before I get into the specific insights, I'm just going to give you a 1.5 minute reminder of sort of high school or maybe college biology. I guess it's probably college biology, and sort of models for evolution, but particularly how they relate to cancer. So the default model for tumor evolution is one of gradualism. It's highly akin to the classical Darwinistic view of species evolution, which basically says that over time, you incrementally, with every cell division, you incrementally accrue mutations. And at some threshold level, you get just the wrong set of mutations that you can give rise to cancer. And of course, this model there's been very good empiric clinical evidence for this kind of approach looking at the evolution of colon cancer. This is a graph, one of the many graphs of the Vogelgram showing progression through adenoma and finally cancer. So, of course, the slope at which you accrue mutations can be affected by a few things. So, for example, if one smokes, the mutational rate can be elevated over time. So therefore, you might cross that threshold earlier in a particular organ than you otherwise would during your lifetime. And another way that you can affect the trajectory is, for example, if you have an early insult. So for example, if you had blistering sunburns as a kid, you now have bumped up your mutational load in melanocytes and maybe are more likely to generate melanoma. But all of these are variants of kind of gradualism. It's just whether the slope is higher than you would expect because of external mutagen, or did you have some insult that pushed you closer to the edge already. But the idea of gradualism as sort of the default and predominant mode of tumor evolution was somewhat turned on its head. Not that it was overturned, but there was a new wrinkle to tumor evolution that came out of a study that was published by Phil Stevens in Cell a few years ago. And this is just as a figure from a review from that. But he came up with he realized that there were certain types of alterations that looked much more radical than kind of the gradualist view of cancer. And in particular, he coined the term chromothripsis to describe a process that happens to chromosomes where there are dozens to hundreds of genomic rearrangements that are highly confined to a particular arm on one or two chromosomes. And in the aggregate, it appeared as though these events would occur through a single genomic event. So rather than have this radical change occur by stepwise incremental selection, it looked like there may have been a single catastrophic event that gave rise to those. And so that basically meant that you have to draw a couple of different models for tumor evolution. On the one hand, you've got your gradualist model, but on the other hand, you could potentially overlay a catastrophic event to that gradualist model that could sort of be gating for a cancer phenotype. Now, in our first study of prostate cancer, which we published, I guess about 2 or 3 years ago now, we saw something that at a bird's eye view looked equally dazzling, but turned out to be very different than chromothripsis. And so I'm not going to walk you through. This is the figure from the paper, which Mike Berger did a great job. I'm sure it took him many hours to make this figure, but it's impossible. It's probably like a rule of how not to make a figure from the standpoint of teaching purposes. So let me illustrate for you what actually is happening in this figure. So if you imagine that you have several different genetic loci scattered throughout the genome, and they can either be on the same chromosomes or very different chromosomes, what we think is happening is that at some point over the course of cell biology, these loci become physically localized. And one way in which they could become physically localized is through transcription. It turns out that there's evidence that the way that we historically learned about transcription, at least the way I learned about it, was you imagine that the DNA is like a rod, and so you have the promoter and the enhancer elements, and the transcription factors come and sit down and they transcribe. But in fact, the genome is not a rod. It's actually, it adopts a fractal globular structure in the genome, so it's very amorphous. It can sort of form and reform, and disparate regions of the genome can physically co-localize. And in fact, there's evidence that transcriptional proteins form hubs or factories within the nucleus to which various regions of the genome go. So if you imagine, on the one hand, that there was physical co-localization of the genome, but on the other hand, that there were some erroneous events. So for example, there was a breakage of several loci followed by misrepair. So you can see is that there's a propagation of breakage and misrepair. What you would get is something that looks like this. So rather than having everything is nicely repaired, but actually there have been a series of rearrangements. Now they would look like chains, but they're not circular chromosomes. They're conceptual chains, closed chain events that have occurred. So with that idea, we realized that this idea of the chromosome chains in prostate cancer is very different than the phenomenon of chromothripsis. So first of all, you don't have hundreds of rearrangements. You tend to have a much more nominal number. On the other hand, the number of chromosomes that are involved compared to chromothripsis is typically more. And the mechanism that is nominated, whereas with chromothripsis, it's thought to be caused by errors in mitosis, for this chaining event, we think it might be involved in errors of transcriptional or perhaps chromatin regulation. I don't have time to go into all the evidence for that, but take my word for it. But already you can see that these look like mechanistically distinct events, and this made us wonder whether or not there needs to be a third model of tumor evolution that needs to be invoked, which instead of being catastrophic would be more punctuated. So this now gives rise to an additional sort of hearkening back to college biology, where Stephen Jay Gould came up with this alternative idea to Darwinism that he called punctuated equilibrium. The idea was that he needed to explain the fossil record, which didn't look like gradual speciation. It looked like discrete fossils. So punctuated equilibrium was proposed to explain that. And this is sort of a graph from an older idea here. So what we wondered is whether or not these chained events are initially telling us that prostate cancer may arise not just by gradual Darwinism nor by catastrophe, but through punctuated equilibrium. So we sort of fell in love with that idea, but of course you have to sort of step back and say, well, can I test this idea and see if it might be true? So you could easily, though, put up a number of postulates that could be addressable by genomic data if this model were true. So one postulate would be that if this is right, we would expect to see these closed chains in a large fraction of prostate cancers. We also might imagine, because it's a series of punctuated events, that some prostate cancer genomes would contain multiple independent chains. So there wouldn't just be one chain, there would be 2 or 3 that were readily evident. And furthermore, we would expect that some of these chains would exhibit subclonality. So they wouldn't be in the predominant clone, they might be in a minor clone, and could we infer that? But most importantly, we expected that many of these chains, if this is a driver model for carcinogenesis, many of these chains should contain known cancer genes. Well, it turns out that the breakthrough to assess this came from an MD PhD student in the lab who recognized that so the way I showed the schema earlier was the way, in fact, we had observed these chains initially, which was the idea that you get basically a clean break followed by repair. But it turns out that a lot of the time, you don't get a clean break. You actually get a break followed by deletion of intervening sequence. And then that deletion is followed by misrepair. So instead of just having clean breaks and rejoining, it turned out that you can have what Sylvan called deletion bridges. And the recognition of the possibility of deletion bridges opened up a door of, wow, there could be many more of these than we thought, because we didn't think to look for deletion bridges. Well, Sylvan, it turns out, so he actually came to the lab to do bench work and to study, biology of a tumor suppressor, but it turned out that he was a math genius, and I didn't know this, but he said, well, this is just a graph theory problem. You just need to, convert this idea of breaks, deletion bridges, and loci to a graphic representations where the rearrangement breakpoints are nodes and the chromosomal deletion segments are edges, and then you can search the graph for sets of connected breakpoint nodes, and you can apply a statistic to determine would these have been likely to arise independently, which would be sort of the Darwinist view, or concomitantly, which would be the sort of punctuated view. So I said, yeah, that's exactly what I was thinking. You should go do that analysis. But it turns out that he was exactly right, and actually what I really told him is you need to bring someone with mathematical skills on your thesis advisory committee. And so he went out and got Francisca Mejor, who is a computational theoretical cancer evolutionary biologist at the Dana-Farber. And so she vetted and sort of affirmed what he was doing. So we were very proud of him for sort of recognizing this. And the reason why this was so transformative is because when he ran his algorithm that came out of this, so he developed an algorithm that he called ChainFinder. And when he ran it, so these are circus plots of prostate cancer genomes, and each chained event has a different color. And they were so prevalent that we decided we needed to give this a name. So Sylvan came up with the name Chromoplexy, so chromo for chromosome, plexy it means to weave or braid, sort of seemed like an appropriate name for these. And it turns out, actually this number is slightly low, 88% of primary prostate cancers out of nearly 60 whole genomes analyzed had at least one event. 2/3 had at least 2 events, and indeed, through data I don't have time to show, but we applied a clonality-based algorithm. We were able to show that in a couple of instances we could detect evidence of subclonality. So one was happening in a miter clone where there was a separate chain comprising the major clone. So now we could go back through our postulates and say, yes, the vast majority of prostate cancers have at least one of these chains. 2/3 have at least two of these chains. There is evidence of subclonality, and now we get to the final question of whether this model could be right, which do any of these chains contain known cancer genes? And the answer was a definitive yes. So here's a representation of an active rearrangement that involved the PTEN tumor suppressor locus, well-known prostate cancer tumor suppressor. But if you are more systematic now, and you take a list of known prostate cancer genes and ask, are they involved in a chromoplexy event? The answer is yes for quite a number of them. So it turns out that the ETS rearrangements, the majority of them are involved. It's not just that you have a temporal fusion, they arise in the context of chromoplexy. So that's something we didn't know before. PTEN is commonly disrupted by chromoplexy, and then here's a series of other known prostate cancer genes that also have more than one chromoplexy event. So this was actually quite exciting. We felt like we had pretty good evidence now for an idea. So, and before I just summarize this, I'll just point out that if you now apply the ChainFinder algorithm to other types of cancer, although it was clearly most prevalent in prostate cancer, you can find evidence in a variety of other solid tumor types as well. So this may be a generalizable tumorigenic mechanism. So this now led us to propose that when we think about tumor evolution, we can't just sort of think of it as a default Darwinistic model, nor is it sufficient to think about it as this is catastrophe. There's a continuum, and so chromoplexy, we think, is reflecting the presence of punctuated evolution in prostate cancer and perhaps other cancers as well. And we thought this was something that was sufficiently interesting that we need to sort of think more deeply about it in the context of what this might mean clinically. So in my last couple of slides, I'll just summarize, and we don't know, but I'll summarize what we think could be interesting about why this particular type of evolutionary history in prostate cancer might matter. So the first thing, and these are questions because we don't know that these are right, but they're worth thinking about. So the first question is, could mechanisms of tumor evolution illuminate new avenues for cancer prevention? So for example, if we understood why chromoplexy happens, what's the error that arises, and you could reduce the prevalence of that error even just modestly, now all of a sudden the slope through which that occurs may be reduced to the point where a lot of patients it'll never happen. So, you know, understanding the basis for this might provide an avenue for chemoprevention. The presence of chromoplexy, perhaps this provides a synthetic lethal opportunity. So for example, like DNA repair defects provide an opportunity for the use of PARP inhibitors. Perhaps there's a vulnerability that arises by the fact that such cells can have this kind of radical event happen. The other question, and we don't know this, is whether or not the presence or the nature, the number of chains could help distinguish indolent from aggressive prostate cancer. And then finally, since we can clearly detect what appears to be an enrichment of known prostate cancer genes in these events, if you see a gene, if you characterize an altered gene and you can show that it has arisen through chromoplexy, is that gene more likely to be a driver event? And so would it be worth knowing some of this when people are designing genomics-based precision medicine type studies? And I'll just point out that if we hadn't done whole genome sequencing instead of even whole exome sequencing, we wouldn't have been able to make these observations. So that was quite exciting from that standpoint. So this is my last slide. We've learned, there's a lot more to learn, and clearly it almost feels like one of these things where you make this great leap, you sequence a bunch of prostate cancer genomes and you realize, well, we're just at the beginning of our understanding. We have no idea how these processes arise, but we do know that we need to know more about them. So clearly what I've told you about is there's genetic evidence for an ETS-negative subset of prostate cancer defined by SPOP mutations and perhaps also CHD1 deletion. This raises the possibility that dysregulated protein homeostasis could be a new therapeutic avenue to explore going forward. There's increased in genomic aberrations between primary and castration-resistant tumors, but I also just told you about the chromoplexy or closed chain rearrangements informing a model of punctuated evolution and appears to be enriched for driver genes in prostate cancer. So I'll just conclude here. Sylvan Baca was the MD PhD student in the lab who led the discovery of chromoplexy. Sylvan also, together with Chris Barbieri at Cornell, led the exome sequencing study that I mentioned earlier. Mike Berger originally discovered the closed chain rearrangements that eventually became known as chromoplexy, and several others on this slide have helped various parts of it. So thank you very much for listening.